We had unknown hormone number 588 by Ashley,
Chris,
Chloe,
and Allie. So this was the data that we had received for the control rats, and we can see here the means and the standard deviations for the control data. This is what we first looked at. So to find these ranges, we added and subtracted the standard deviations from the means for each body part. This gave us ranges for the control and experimental data for each body part. After that, we compared the ranges between the experimental rats' body parts with the corresponding control rats' body parts to see if they overlapped. If the data ranges did overlap, we could tell that the data was too similar, and because of that, it was not significant. On the contrary, if the data did not overlap, we could tell that these body parts were very different. And then this here is just our data for our specific experimental rats. Upon reviewing the control and experimental data, we could see that the uterus, the thymus, the tibia, the pituitary gland, the prostate, the seminal vesicles, and the body weight had a difference compared to the corresponding body parts in the control rats. The uterus, the thymus, the tibia, and the pituitary gland had the largest statistical significance. We could see all this because the data ranges of the experimental rats' body parts did not overlap with the data ranges of the control rats' body parts. Along with these, there was a body part that was to be of interest. This meant it did overlap, but by a very small amount. This was the ovaries. This was a body part that we did not completely eliminate when deciding on our hormone, but did not entirely base our decision on either.
then the three possible hormones we decided on were somatostatin, HCG, and estriadol. When we had to narrow down the possible hormones to just three, we decided upon somatostatin, HCG, and estriadol, like I just said. We decided upon somatostatin for a possibility because it affects the endocrine system and the bones. In our experimental data, our tibia width was smaller, which tells us some of the bones would be smaller thanks to growth hormone inhibiting hormone, also known as somatostatin. When considering HCG, we believed it could be this hormone because you get this hormone when you are pregnant, but this really shouldn't affect the tibia. Lastly, we thought our unknown hormone could be estriadol because it is related to the ovaries and is associated with menopause. We found this to be very significant because our data showed a huge change in deviation regarding the ovaries and uterus compared to the control data.
Upon narrowing it down to our three possible hormones, we then felt prepared to begin requesting our test results. The first test we requested from Professor Cotty was physical appearance. We requested this test in order to check for mammary growth, overall size increase or decrease of our rats, and also to be made aware of any changes in behavior. Once we received our results, we were then able to narrow our possible unknown choices down to HCG and estriadol. We felt comfortable eliminating somatostatin as a possibility due to no change in the overall size of our experimental rats. At this point, we knew we were headed in the right direction with HCG and estriadol. The results that were most helpful in leading us to these choices are as follows: development in mammary glands, slightly elevated time spent feeding, more time spent nest building, and lastly, they showed mild defensive behaviors of their nesting areas.
The second test was the glucose tolerance test. Somatostatin causes a lower blood glucose level. Therefore, the glucose tolerance test was vital in determining whether or not somatostatin was our unknown hormone. As you can see in the graphs provided above, in the control rat, it showed levels of pre-diabetic glucose levels. Before eating, the control group rested in a normal range, but 30 minutes to 1 hour after food consumption, their levels spiked to pre-diabetic and then back down to normal after 2 hours. In the experimental rats, after 30 minutes, their glucose levels reached pre-diabetic, but they continued to grow almost up to the diabetic range, being only 5 milligrams off. It eventually came back down to the normal range, but if somatostatin was our unknown hormone, the blood glucose levels in the experimental rats would not have gotten that high.
And the third test we decided on was an RIA test in plasma for estriadol. A radioimmunoassay in plasma for estriadol was completed, and we found that the experimental rat but a significantly smaller number than the control. If you find a smaller amount of radioactivity in the experimental rat than the control rat, that means that there is a large amount of the tested hormone in the sample. Given this information, it is shown that there is a high level of estriadol in our experimental rat. Then the two hormones we eliminated from our list were somatostatin and HCG. When looking back upon the data from the different tests that we received, we decided to eliminate somatostatin and HCG from our possible hormone list. When looking back upon somatostatin, we found that our experimental rats had large levels of glucose. Somatostatin is produced in the pancreas and inhibits the secretion of pancreatic hormones like insulin and glucagon. If our rats had high glucose levels, then somatostatin couldn't be a possibility. Also, somatostatin doesn't affect the reproductive organs, which is one of the largest changes in our data set. When looking back upon HCG, we know that it was produced during pregnancy and it doesn't affect the growth of bones. HCG and estriadol are similar in a lot of ways, and to figure out which of these hormones we had, we decided to ask for a pregnancy test for our third and final test. We found out that both the control rats and our rats were not pregnant. This shows us that it couldn't be HCG due to the lack of pregnancy.
So we decided that our hormone was estradiol.
In females, estradiol acts primarily as a growth hormone for reproductive organs, including the vagina, the fallopian tubes, and the cervical glands. During the menstrual cycle, follicles on the ovaries secrete estradiol. This secretion triggers a surge in luteinizing hormones and in turn induces ovulation. Estradiol is also needed to support adequate bone growth as well as help us maintain healthy bones and joints. Lastly, estradiol's function in males is to maintain a healthy libido and aid in maturation of sperm.
And then this is one of our works cited pages. And that's it.